Okay, our next speaker will be Virgil Reese.
Thank you.
About the death.
Well, first of all, I want to say how much I've enjoyed the other talks. You know, I started this idea I have not thinking it was going to have anything to do with the reason for sex to occur in nature, but I think you said 20 different, I'd make it 21 different explanations for why sex might exist. This is, it's an idea that's either nothing or it's pretty important, I think, the way I look at it, because it really does offer a novel explanation for why sex would exist in nature, independent of the others that have been mentioned. Not that any of those are necessarily not also beneficial, but I think this one, you'll see, if it's true, offers a powerful reproductive advantage to sex. So the question I ask is, does meiosis facilitate an error correction mechanism that can bias gene conversion in favor of existing wild-type alleles and against new mutations? Well, my answer to that's obviously going to be yes, it can. It was a surprise to me to realize that. But the title, I kind of regretted right after I did it. I didn't really know much about gene drive until recently, and I realized that my mechanism is really a subset of gene drive, but it's a little different. So gene drive is a homozygosity enforcing pathology, basically. It initiates homologous recombination and then results in gene conversion that favors the initiating, therefore, gene drive harboring strand. So this is invasive. This error correction mechanism that I propose is also homozygosity reinforcing, and it also involves homologous recombination that's induced by what I'll call a dangerous allele. In this case, it's not necessarily pathological, but potentially. The key difference between what I'm proposing and gene drive is that in this case, the recombination is going to result in gene conversion biased in favor of the non-initiating allele, which is the more common thing that you're going to find in nature. I mean, in the case of double-strand breaks or any kind of damage where you have to go to homology to fix it, you obviously don't want the initiating strand, the one that says, help me, I've got something wrong, to be the donor strand. So this would be gene conversion favoring the stable strand that doesn't have this potential mutation, and therefore will likely remove damage rather than spread it. So the first process, pathological gene drive, is invasive, whereas what I'm proposing is a defense against invaders. So this is an error correction mechanism, and I'm just going to say a lot of my talk is on here. You know, don't feel obliged to read it. It's my notes, since I don't have I don't memorize things very well. So what I'm proposing is an error correction mechanism that enables sexually reproducing organisms to create haploid germ cells with a lower than chance share of the recent mutations to highly conserved regions of their autosomes. And I say to highly conserved regions. This obviously wouldn't happen across the genome. What I'm talking about is, you know, we have regions highly conserved, ultra conserved, where we share the same sequence as bacteria or petunias or flies. So these are areas where if one of your two chromosomes differs from the other, there's a pretty good chance that one of them is wrong. The only problem is which one. And the answer to that question isn't seen to be clear. So this mutation in a highly conserved region, a SNP, you know, single nucleotide polymorphism, is a reliable signal for the presence of mutation. And I'll also be talking later about the same logic can apply to larger structural variations. But this information is generally seen as unhelpful to the organism simply because it has no way to discern which of the two non-matching alleles to suspect. I'm arguing that sexual reproduction introduces a way for not for that organism to know the answer to that question, but for that organism's offspring to discern an answer to that question. So the key, if I can, is to create within the primordial germ cell line, or in the germ cell line, some sort of transgenerational epigenetic signature at or near this heterozygosity that would basically just mark both alleles as suspect. Again, your genome knows that there shouldn't be a difference there, but it can't tell what to suspect, so it just marks them both and says, I'm not sure, something seems fishy. So this information then can be useful once a new reference chromosome becomes available. Well, how are you going to get one? You know, I say there's a good way to do it. Fertilization, sex, provides a new reference chromosome. So you've got this mark. When either of these two suspect alleles, and presumably one of them, will be paired with a new homolog, any persisting heterozygosity, now it could be that there isn't one. You know, this one that was marked as suspect may have been the good one. But if it's not, you see heterozygosity. And this will not just signal, again, the presence of a probable mutation in the new organism, but it will also tell you who the likely culprit is, which is the one that was previously marked as suspect. Excuse me, I forgot to bring my water over here. I can't talk for more than 30 seconds without taking a sip. So that's the key. I think you're sort of getting where I'm going here a little bit. So error correction could then occur in the new organism, and I propose not in the zygote. This is where you'd want to do the least surgery on the cell. This is a very fragile place, but just in the germ cell line of the new zygote. This suspected allele will have had a mark associated with it, and then during meiosis, it will be what induces meiotic homologous recombination. Not necessarily crossing over. I'm just talking about, about 90% of recombination in meiosis is actually non-crossing over. And so all I'm really looking for is a single-strand exchange or branch migration. Here, the suspect SNP can be directly paired via single-strand exchange with a new homolog, which in the case of an actual mutation would create base pair mismatch in the new organism. This is just pictures of what I just said. So you've got the green, which represents the mark that I'm suggesting. S is suspect, W is wild type, okay? So you got one chromosome that had already been marked as suspicious. Fortunately, when it finds a new chromosome after sexual reproduction, the other one is more likely to carry the wild type allele. The mark is what initiates recombination. Again, this is not crossing over, just strand exchange. And after strand exchange is resolved, you end up with a base pair mismatch on both chromosomes. Now, the standard theory says you got a base pair mismatch. One of those is going to get replaced with the other, and if it's random, half the time you spread the mutation, half the time you get rid of it. Why go by chance? If you already knew that one of them had been suspicious, you're just going to, and of course in this case, you already know that the gene conversion is biased against the initiating strand normally, unless you have pathological gene drive active. The tendency is for the non-initiating strand to be the donor strand. So that will automatically tend to simply replace the suspect allele, and basically you rid yourself of a mutation. Now again, I think what's remarkable about this is it requires sex to do it, and it provides a pretty powerful benefit. And I suppose that this could be a benefit that explains why sex is common in nature. That's getting ahead of myself, but that's my argument. And again, it's because mismatches are typically resolved by gene conversion that's biased against the initiating strand that this will naturally tend to replace suspect alleles, which half the time would represent a mutation, assuming we're talking about a highly conserved portion of the genome, and it would replace them with what's more likely to be the ancestral or the wild type alleles. So it's a neat little trick if you can do it. Now, I'm not a scientist. I've passed this off to many biologists over the years. Most of them have said, wow, I've never heard of that before. It'd be a neat thing if nature could do that. I don't know, I'm kind of suspicious. But my attitude is, if it's a neat trick, nature probably would have discovered it. And I finally got one scientist who took me seriously enough, Donald Forsythe at Queen's University, who tried to get me to write up a better paper because he had reviewed my original paper that I sent to the Journal of Theoretical Biology. Rather negatively, he apologized a couple years later when the idea stuck in his head and he couldn't get it out. So he finally, after I just refused to do the work and write the paper, he wrote it himself and asked me to be co-author. And of course, I don't know, I ended it odd and said, oh, okay. So anyhow, we just got recently published in Biological Theory. I've got some paper copies if anybody wants to hang out at my poster later. But anyhow, so these are just 3 basic steps. I'm going to summarize the steps. One, okay, so identify, mark, and then remove rare SNPs. Homolog pairing in meiosis enables some fraction of the genome's highly conserved DNA to be scanned for isolated SNPs. The details of that didn't go into, won't go into. I think there's reasons to believe that it would be hard to do more than a small portion of the genome per generation, which partially can explain why we still have mutations. This isn't a cure-all. And at or near one of these identified SNPs, both chromosomes will receive an enduring epigenetic mark in transgenerational. So that whichever one of the 2 alleles is paired with a new homolog after fertilization can then be treated as suspect. Then in the next meiosis, the suspect allele, or SNP in this case, induces homologous recombination, which is then repaired by, after its first strand exchange. Gene conversion resolves any mismatch in favor of the safer correction template strand. So that's it in a nutshell, and that's where it remained in my brain for actually several years. But Forsythe started talking about different types of mutations, not just SNPs, and it occurred to me that error correction of this nature doesn't have to just work by gene conversion. It doesn't have to just work for single nucleotide polymorphism. So the same argument is going to now be made for larger structural variations, deletions, duplications that might also be suspect. So you have the same 3 steps. And this is my artist's rendition of synapsis, which is an interesting phenomenon, unique to meiosis. It doesn't occur any other time in any other cell division. And so I think that one of the things it does is it makes it more easy to bring into appearance any large structural differences, because it's basically pairing the genome all the way along, kind of like a zipper thing. And of course, the loops are pieces of DNA that if something's missing, it's going to be obvious, and if there's too much there, it's going to be obvious as it tries to pair up things. So I suspect that, well, I use the word suspect, that if there's a place where homology does not exist, it should be markable. This would be a good place to find it. And then once that happens, now I've used red as the color to represent some place that was, and I exaggerated the length of, obviously, homologous chromosomes don't generally vary that much in length. I'm just trying to point out this might have been a large addition. The other red on the other chromosome could be a deletion. So you've got the red areas are suspect. Crossing over in this model would not just be for some of the other traditional explanations for crossing over, but it will create at least some recombinant chromosomes which have been partially depleted of these areas, these structural variations that had already previously been marked as suspect. The red means it has been marked. So I have SSV, which stands for suspect structural variations. And so the one, if you can see the black circle, that one there is has the two suspect areas from the parental chromosomes have been removed from at least one of those 4. And of course, in meiosis, you have this interesting thing. Instead of just starting with a diploid cell, and then all you need is a haploid cell, why not just cut it in half? First they double it, then they cut it in half. I think that makes more sense if you think of it in terms of sorry, I'm getting ahead of myself, and I'm also losing my script there, okay. So my argument here is that the independent assortment of chromosomes isn't truly random. What it's doing is it's segregating towards one. Oh my goodness. Okay, so you get the idea. This is well, that was fast. So basically, I'm arguing that in egg cell production, you've got the polar bodies which eliminate 3/4 of all the products, and so you are eliminating. If you're segregating the repaired ones together, you're going to end up with germ cells that are mostly made of repaired cells. So again, this is a 3-step process. The same 3 steps. During close association between homologs, you get identification of suspect SNPs. Then once they're suspected, you make now this would be a different signature because the cure is different. It's not gene conversion, but crossing over. And then in the subsequent generation, you get the repair. So why do we still have mutations? We still have lots of mutations, and I'm arguing that, so this isn't something like a cure-all for mutations, but it's more likely to be the case that it can remove mutations until they become harder to find. If there's a higher mutational load, there's a thicker rate of them, they're easier to find. You can't reduce it down to 0. I'm just going to skip that because I want to get to a couple things. Falsification. I'm just going to say that there's 2 obvious predictions. Isolated SNPs in highly conserved DNA regions must be inherited at lower frequencies and expected under strictly Mendelian models. My argument is that this is going to be a fairly small deviation because we have, humans for instance may have about 800 slightly deleterious alleles. If we were removing 1% of that per generation, that would be as many new mutations, deleterious mutations as we typically have. So I'm talking about a small deviation, but nevertheless it has to be found, and so that's a strict prediction. The same thing with structural variations. They have to be found to deviate less so. Now I think that the explanatory potential for this is obvious. I'm just going to say sexual reproduction has a new potential explanation, asexual reproduction. I don't have time to go through all those, although there's nobody coming after me. So if anybody would like to let me go just a couple more seconds, I would love to do so. But, well, let me. Is that okay with you? People, feel free to leave. I want to talk about just. We talked a lot about sexual reproduction here, and I've learned a lot from it. But I want to point out that in this particular case, even though sex may help remove mutations by shuffling around, this is a far more powerful potential strategy. But more importantly, and I'm going to just skip the asexual stuff, crossing over has a completely different possibility. But what I want to point out is that unlike natural selection, this error correction mechanism has potential to remove deleterious mutations even before they become fitness burdens. And I think that that's an important point, and I'm just going to skip to my, boy, I really went over, didn't I? It just repeats the same thing. I have one question that I want to ask, and I think it's a good question for anybody to ask. How likely is it that life would have never found this simple way to combine existing processes capable of significantly reducing the burden of purifying selection and thereby greatly increasing levels of genome complexity that species can sustain? Well, I better leave it at that. I'll skip the other thanks, but I especially owe thanks to Dawn, of course. And I will be hanging out my poster if anybody does want to discuss it. And if anybody has any questions they want to ask me now, they can.